Hi everyone, I hope you are all well. It's a great honor to give a talk at OHBM this year. And today I will talk about the research that I conducted with Byeol Kim and Jessica Andrews Hanna. How do you feel now? And what's in your mind? What comes to your mind? So our mind never rests. Thoughts and feelings come and go. And our mind usually wanders around here and there. So it's known that people spend almost 25 to 50% of their time for mind wandering, suggesting that the mind wandering is an actually important part of our life. For a deeper understanding of mind wandering or spontaneous thought, I like to use a network representation where the nodes are concepts or semantic elements which serve as building blocks of spontaneous thought, and the edges are the associations among them. Some nodes could have negative valence, while some could be positive. And some could work as a strong attractor, like the black hole, so that an individual ends up having the thought very often. And some could be too sticky to get out of the thoughts. Then we can conceptualize the mind wandering as a random walk on this network. With this in mind, we devised a new fMRI task based on a free association method named Free Association Semantic Task, FAST. It consists of two parts. The first part was the word generation task, where we provided a seed word for each run, and we had 4 runs in total, so 4 seed words, and asked participants to generate a word train of 40 associations. We gave only 2.5 seconds for each association to minimize the censoring. After participants generated 40 words in a row, we conducted a word thinking task where we showed 2 consecutive words generated by themselves and asked them to think about the personal meaning of the second word. That was the target word. And after the scan, we showed the words again and then asked multiple questions about them, including the emotional valence, how positive or negative, and how self-relevant, and relevant time period from past to the future, and how vivid, and also lastly, how safe or threatening the word is to them. In sum, we developed a new task to understand the temporal dynamics of spontaneous thoughts and emotions, and also the brain representations of affective experiences induced by self-generated thought. To show what we could do with the data from this task, I'm showing one participant's example data here. We can visualize the free association trajectory on the affective dimensions. I'm showing only 3 dimensions. In this example, the participants talked about Bitcoin, suicide, fail, bubble, and so on. So time we conducted this experiment was around 2018 when the Bitcoin crash happened. And this is a network representation of one participant's data. The arrows here show the trajectory, the direction of the free association, and the color indicates the valence ratings. So this particular example shows 3 large clusters, and the first one was about family. So this participant seemed to have a brother and fought with him, and parents were in the middle, but each family member like mother, father, grandmother, all positive. So you can make a guess about the family dynamics from this data. The second cluster was about school and also objects and places, and they were mostly positive. And the third cluster showed a top node that was sadness. The node is connected to loneliness, disappointment, regret, worries, and also girlfriend. So these example data really highlight that a participant's response patterns to the task can provide rich information about the individual in terms of thought topics and affective tones related to them. And there are so many possibilities in how to analyze these data, including dynamic modeling, for example, Markov chain analysis and network modeling and predictive modeling, or combining it with natural language processing. So which are all what Jess and I are currently trying, but today, because of the time limit, I want to focus more on the fMRI data. So we particularly focused on the brain activity patterns during the word thinking period, and I want to start with showing the basic contrast map. So during thinking about the words, the default mode network regions, including medial prefrontal cortex and some subcortical limbic areas, including hippocampus and amygdala, were activated. However, unexpectedly, the visual cortex was deactivated even though the stimulus was given visually. To better understand this map, we conducted the clustering analysis based on the temporal profiles of brain activity using an FIR model. The K-means clustering analysis suggested the 4-cluster solution, and we further combined the cluster 1 and 2 that showed similar temporal patterns and also similar locations. So the first 2 clusters included VM and DMPFC, TPJ, IFG, and so on. These regions, these clusters, showed a delayed activation pattern, these 2, green and yellow. And if we do the decoding analysis using Neurosynth, a large-scale meta-analytic database, these regions were best explained by the terms like autobiographical, remembering, emotional, and memories. So we named this cluster as meaning related. The third cluster, red here, that included the somatomotor system like S1, S2, and also the insula cortex. This cluster showed a large deactivation right after the word onset and slow increase toward the end of the word thinking period. And it was best explained by the term including somatosensory, stimulation, and touch and pain. We named this cluster as sensory motor. And the fourth cluster that included the visual cortex and periaqueductal gray here. And this cluster showed a transient activation right after the word onset and then a large deactivation. So this cluster was best explained by the term including visual, sighted, navigation, and so on. So we named this cluster as stimulus driven. We then examined the regional time scale of internal state transition using this word thinking period data using the hidden Markov model implemented by Baldassano et al. So in this study, using a naturalistic movie viewing task, they showed that the early sensory regions had a fine time scale while the default mode network had the coarse time scale of event representation. And if we use our cluster solutions, we could say that clusters 3 and 4 will show a fine time scale, whereas clusters 1 and 2 will show a coarse time scale. So this is the whole brain time scale pattern of state transition based on the word thinking period data. And when we use the cluster ROI masks, our results show that the clusters 1, 2, the meaning related cluster, green color here, covered both fine and coarse timescales of state transition, providing different patterns of results from Baldassano et al. Maybe because we use the self-generated endogenous stimuli instead of exogenous stimuli. We then wanted to develop whole brain pattern-based predictive models to predict different affective dimensions using principal component regression. And the cross-validation results show that we were able to predict only the level of self-relevance and not others. And this is the predictive map for the self-relevance, and these regions are the ones that make reliable contributions to the prediction of self-relevance ratings. And consistent with the meta-analytic map for self versus other, the medial prefrontal cortex region showed positive weights along with other regions like nucleus accumbens. And for the prediction of valence rating, given that we couldn't predict the valence score directly from the trial level images, we used the divide and conquer strategy because we had a hypothesis that depending on the level of self-relevance, maybe the valence representation in the brain could be changed or warped. So we divided the data into high versus low self-relevance trials and then predicted the valence ratings, and we found that we were able to predict only the valence ratings with low self-relevance trials. not for the high self-relevance trials. And this is the valence model for low self-relevance trials, and it shows the negative to positive gradient in dorsal to ventral medial prefrontal cortex, consistent with previous literature. And we tested the valence low self model on an independent test dataset from Luke Cheng's 2015 PLOS Biology paper, in which they showed the IF's pictures to evoke negative emotions in participants. So importantly, our model worked in these two different datasets, their training data and testing data. Does the valence low self model seem to work even in independent datasets? But why valence high self didn't work? And our reasoning was like this. The models so far were designed to work for population level prediction. So that is one model for all people. And it didn't work for high self-relevant trials, maybe because valence representations in the brain become idiosyncratic when processing highly self-relevant information. So to examine this idea, we developed individualized predictive maps of valence ratings with high and low self-relevance trials separately. And even though the model performances of these two models were comparable, the between-subject variability of predictive weights was much higher for the high self-relevance trials, supporting our idea that when we process highly self-relevant stimuli, our brain representations of valence become variable across people. So in sum, we developed a new fMRI task that can provide rich information about personal thought topics and their temporal dynamics. And the brain representations and dynamics induced by the task were quite different from tasks with exogenous stimuli, and the brain representations of emotional valence became variable across people when the stimulus was highly self-relevant, suggesting the importance of using self-relevant stimuli for studying emotional brain. With that, I want to thank the funding sources and also to our Kokwan lab, and Byeol Kim did the most of the work, and Jess has been a great collaborator for the project. Thank you for your attention.